Ribonucleic acid, also what we call the RNA, is a nucleic acid that is present in all living cells. RNA is most often a single-stranded molecule that has a backbone mode of alternating phosphate groups and sugar ribose. One of the most evolutionarily conserved properties of RNAs is the RNA modification. RNA editing occurs in all living organisms. It is a molecular process through which some cells can make these changes to specific nucleotide sequences within an RNA molecule after it has been generated by RNA polymerase. Since pre-mRNA has to go through some modifications to become a mature mRNA molecule that can leave the nucleus and be translated, they go in the process of splicing, capping, and addition of a poly A tail.
The capping process replaces the triphosphate group with another structure called the cap. The cap is added by the enzyme guanyl transferase. This enzyme catalyzes the reaction between the 5' end of the RNA transcript and the guanine triphosphate or GTP molecule.
After the process of capping, the process of tailing is next. The process of tailing is the post-transcriptional addition of non-templated nucleotides to the tree end of various types of RNA molecules. It is a highly conserved process that modulates their stability and activity in multiple organisms and plays a crucial role in regulating gene expression. So basically, the process of tailing is all about providing support and stability before the RNA goes to its next process, which is de-splicing.
Splicing is a fundamental process in the biogenesis of eukaryotic messenger RNA molecules. It involves the removal of non-coding introns and the joining of coding exons to form a continuous, functional mRNA molecule that can be translated into a protein by ribosomes. The splicing process is carried out by a large, multi-subunit complex called the spliceosome. The spliceosome recognizes the consensus sequences at the boundaries of exons and introns called the 5' site and 3' site respectively. It then cleaves the phosphodiester bond between these sites, releasing the intron as a separate molecule and joining the exons together through a new phosphodiester bond. This process is highly regulated and can be influenced by various factors such as alternative splicing events that generate multiple mRNA isoforms from a single gene and splicing factors that bind to specific sequences in introns or exons to modulate splicing efficiency or specificity. Overall, splicing is essential for the proper functioning of eukaryotic cells, as it allows for the generation of diverse proteins products from a limited set of genes, and helps to ensure that mRNA molecules are efficiently processed and accurately translated into proteins.
To summarize it all, RNA modification or editing happens to form a mature translatable messenger RNA or known as mRNA. The modification undergoes in 3 processes: the capping, tailing, and splicing. Wherein capping, a protective cap is added to the prime 5' end of the mRNA, and tailing includes adding a string of adenine to the prime 3' end of the mRNA molecule. While splicing includes the removal of introns and joining of extrons together to form a mature mRNA. And that's it. That's where our RNA modification ends.